Okay, I'll get this lecture started for today. So, before starting, I'll just like to clarify two things: whether my is my screen visible and whether my recording is audible to you guys. Just can you just type it in the chat box if you can't say it say it out loud. If you can't open your mic. Okay, I am assuming that my screen is visible and my audio is audible to everyone. So, welcome everyone once again to week two. Sorry, this is I accidentally put this as week one. Welcome to week two of this TSF session for the course Bioengineering and Interface with Biology and Medicine. The course code for this course is NOC 25 BT05. And hello, and just to give a formal introduction about myself, continuing from last week, I am Arjun and I am a third year PhD student at Indian Institute of Science, Bangalore. And this is my mail ID if anyone wants to contact for any doubts related to this TA session. So, without further ado, I'll just jump right into the contents of this course. So, the plan for this TA session is kind of simple. So, I'll just go over a brief recap of whatever happened last week in week two of the lectures of this course. And after that, I'll have a short doubt session, following which we'll discuss uh, previous year's assignment questions for week two. After which I will have one more session, and we will conclude the session after that. So I'll start with the week's content. So this week, the contents of the week mainly focused on a basic introduction into some molecular biology and molecular biology techniques, and the course instructor, Professor Sanjeev Srivastava, started by explaining about nucleic acid and the central dogma, and he explained about how genetic information is stored in DNA, and this DNA can be used to synthesize RNA, and which in turn can be used to synthesize proteins, which forms the central dogma, and where DNA, RNA, and proteins are used at different stages for different purposes. DNA is used for storing genetic information in living organisms, while protein is used to synthesize amino acids are combined together to form proteins, and proteins are used to perform metabolic functions in our body. And we also saw the difference between how deoxyribonucleic acid is formed, where and how nucleosides are formed, wherein we have a base, and the base can be ribose or deoxyribose depending on what kind of nucleic acid are we dealing with, whether it's a DNA or an RNA. If it's a DNA, the base will be a deoxyribose, whereas if it's a RNA, the sugar will be a ribose sugar. And the sugar, if it's a conjugated with uh, ba- uh, sorry, is a sugar if it's conjugated to the base like adenine, guanine, thymine, cytosine, it forms a nucleoside. And the nucleoside, if it's conjugated with a phosphodiester bond, it's called as a nucleotide. And multiple nucleosides conjugated together forms nucleic acid. And we also saw how DNA is packaged, how DNA is not linear as we see it is. DNA is packaged into chromosome through histone with the help of histones. DNA is wrapped around histones, forming a histone octamer. The histone octamer is further compressed to form chromatin, and this chromatin is further compressed to form a chromosome. And following that, we also saw more about gene regulation, and we saw an example of gene regulation, which is the lactose operon, where we saw about promotion of promote, where we saw different facets of gene regulation such as promoter repressor and how these promoter and repressor interplay in different kinds of regulation such as positive regulation or negative regulation to produce different proteins at different stages. And the course instructor also went on to explain about several several milestones regarding to DNA discovery, how DNA structure was found using X-ray crystallography, and how the double helix structure was found, and how when the genetic engineering techniques helped in the advent of gene therapy, and how cloning was helpful in creating this first synthetic sheep, that is Dolly, etc., etc. And after that, we saw went on to discuss about DNA tools, where we initially discussed about plasmids and cloning vectors, and we discussed about one such example, PBR322, which had a couple of antibiotic resistance genes, and which had a couple of other genes which helps to um, which helps in the selection process. And we also saw a brief introduction to the steps of DNA cloning, which involved which were mainly differentiated into three different steps. DNA cloning. The first step would be the first step would be cloning. The second step would be transformation, and the third step would be selection. So the cloning or the cloning would be the process where we could see the formation of the recombinant DNA molecule. So if we have a plasmid, the plasmid would be uh, cut into two or three or multiple parts depending on our choice by use of exonuclease. Oh, okay, then I think it's just a disturbance. So in the first step, which is cloning, we basically introduce our target gene to the plasmid. So what we do there is we have a couple of um, exo, exo, we have a couple of exonucleases, and we have a couple of splicing sites in the plasmid through which we introduce gaps or cuts, and then this gaps or cuts will be sticky, so there will be some couple of overhangs. So after that, what we do is we introduce our target DNA, and we add something called as a ligase, which is used to bind the target DNA with this overhangs of the target DNA with the overhangs of the plasmid. So this is in the formation of a recombinant DNA molecule. And the next step is transformation, where this recombinant DNA molecule is introduced into the bacteria, and the bacteria using this plasmid will produce multiple copies of this target DNA. And to confirm whether our product has been generated or not, we select the bacteria which has only the target DNA. So along with the target DNA, we'll also have specific sites for antibiotic resistance as well. So we plate these kind of bacteria in agar, and then we add an antibiotic so that the cells which don't have resistance to this antibiotic will eventually die, and we can finally select the bacteria which has this antibiotic resistance. And finally, we also saw how we could test the formation of this target DNA using agarose electrophoresis. Agarose electrophoresis is a technique using it's a charge based technique where you uh, load your sample or your target DNA on one in several wells, and then you run the gel across the electric gradient, and you compare the molecular weight of your target DNA with the standard molecular weight ladder, and then you compare the molecular weights between the standard and the sample to check whether your target DNA with the desired molecular weight has been successfully incorporated into the bacteria or not. And finally, to conclude the week, we also saw a detailed overview about polymerase chain reaction, where we saw the different components required for the uh, polymerase chain reaction, namely Back polymerase, which is which is isolated from the organism Thermus aquaticus. We also saw the reason why we are using this Thermus aquaticus Taq polymerase, since it can withstand high temperatures. We are using it, and we use high temperatures to denature the DNA, which is the one of the first steps involved in PCR. We have three steps in PCR, which is denaturation, annealing, and extension. And apart from the template strands, we also use primers, which are short complementary sequences to the DNA template strands, which are used to bind to the template strand and which are also used in the process of annealing and extension. And apart from the primers, we also have dideoxynucleotides, which is adenine, guanine, thymine, and cytosine, the deoxy forms of them. Those are used in the process of extension, where we keep on adding nucleotides based on the complementarity of the template strand. So that's all about the short recap that happened in this week two. We had discussed about central dogma, nucleic acids, and we discussed about cloning and polymerase chain reaction. So I'll keep this room open for the next 5 to 10 minutes. After that, which we can discuss some of the questions and answers from the previous assign previous year's assignment questions of week two. So there are a couple of questions from week two of previous year's assignment. So most of them are like problem based. So we can go over each question, and I'll give you some time to discuss over them, and then we can try to solve the questions one by one. So I'll keep the room open for another 5 minutes. From 3:15, we can start the discussion of the previous year's questions. If you have any doubts, you can open your mic and ask, or you can also type it in the. Oh, sorry. So, sorry. If you have any doubts, you can open your mic and then ask any question. Or if you can't open your mic due to network issues, you can always feel free to type your questions in the chat box. Okay then. So if no one has any doubts, I'll move on to discussing the problem statements. So we'll go over each question. I'll give like 5 to 10 minutes for each question, and we'll just see if I'll give you guys time to discuss all the questions as well. We can you guys can use the chat box as well. So I'll give you guys 5 minutes for every question, and then we can solve the problems one by one. Yeah. So first question, I'll read out the question loud as well. So Bhavana is a very bright research scholar. She extracted DNA sequence from unknown bacteria from the waters of Prayagraj Sangam. Now she wants to amplify the DNA for sequencing. Please go through the setup of PCR set by Bhavana carefully. So in the last like the fourth or fifth lecture we saw about PCR. We also got to see a live demo of PCR by one of the instructors and students. So this question is about PCR. The reagents used for PCR in this experiment are tag DNA polymerase, PCR grade water, primers, dideoxynucleotide phosphates, DDNTPs mix, PCR tubes, thermal cycler, PCR tube. Yeah, PCR tubes is repeated twice, so it's okay. And cycling parameters are 94 degrees for 1 minute, 64 degrees Celsius for 2 minutes, and 72 degrees Celsius for 3 minutes. Choose the correct statement among the following. So, you guys already, I presume that you guys already have went through the lectures. So, here we already know what the reagents are for. So the tag DNA polymerase is a DNA polymerase which is used to uh, which is used in extension, and we need PCR grade water as usual. Primers are complementary sequences which are used to bind to the complementary template sequence, and we need dideoxynucleotide DDNTPs for extension, and we need a thermal cycler because each step in PCR requires needs to be operated in a different temperature, and we need PCR tubes for these reactions to be performed. So they have asked us to choose the correct statement among the following options. So the first option is the PCR reaction would fail as the extension temperature needed to be set at 60 degrees Celsius. Second option is the PCR reaction would fail as the denaturation temperature exceeds the melting temperature of the primer, and the primer would also denature. Third option is the PCR reaction would fail as stacked DNA polymerase are not effective enough. The fourth option is the PCR reaction would fail as DDNTPs won't allow to incorporate other nucleotides and the extension would stop. So, which option would be the correct option? So, there is a small uh, error in this question. So, you need to find that out, and you also need to uh, tell why that will be the answer. So we saw in the recap as well that what are the reagents required. So for the way that we'll be going over this answer is go over the options as well, see if it makes sense, and if the options do make sense, if you can't find the error in this question, which is the answer, then you go over the reagents one by one and see if they really make sense in being used for PCR, or do they need to be changed slightly. So does anyone have an answer for this? I'll give you time guys till 3:20. So we are 3:18 now. So the question is pretty. It's not simple. It's straight. It's not simple and straightforward. There is a small twist in this question, and that's what you need to find out. And you just need to rationally think why this won't happen. So we talked about DNA, how DNA is formed, how nucleoside and nucleotide, you can also think it in this way, how nucleoside and nucleotide will be formed, and what will be the essential thing that will be involved while forming bonds between one nucleotide and the other succeeding or preceding nucleotide to form the nucleic acid. So which one of the parts will be helpful for this bond formation, and what is missing in this? Mrunmay Jyoti, if you like, I don't know whether the thumbs up is for a doubt or if you want to answer something, you can open the mic if you want to answer, or you can type your answer in the chat box if you feel like you are confident about an answer. Okay, so I'll go over the options one by one, and I'll discuss why this is possible, why whether this is possible or not. So the first option is the PCR reaction would fail as the extension temperature needed to be set at 60 degrees Celsius. So this option is pretty debatable. So if you see the cycling parameters, there are three different temperatures: one at 94, one at 64, and one at 72. So 94, we already know that is the denaturation. If something is at 94 temperature degrees Celsius in a thermal cycler, then that would be the denaturation temperature. So the double-stranded DNA will break into two single-stranded template strands, and the amplification will happen at 94 degrees Celsius. 64 degrees Celsius will be the annealing temperature where the primers will bind to the template strands. So that is also a proper temperature. The annealing temperature, you guys know from the from the week's lectures, that it depends on the melting curve. We do a melting curve analysis and we find the melting temperature, and that which melting temperature also depends on the GC content, the guanine and cytosine content of the primer, and the melting temperature can be always okay if it is 1 or 2 degree above or below the ideal melting temperature. So if they have a melting temperature of 64 degrees Celsius, extension temperature of 60 degrees Celsius is permissible to an extent. 72 degrees Celsius is the temperature where the extension would happen, then that will be the normal extension temperature for any Taq polymerase based experiment. So the first option will be automatically rejected. The It will be a wrong answer. The second option, PCR reaction, would fail as the denaturation temperature exceeds the melting temperature of the primer, and the primer would also denature. So denaturation is just a process where the double-stranded DNA breaks into single strands. So since primer, you already have forward primer and reverse primer, which are single strands in nature. So after that, they can't denature further in this temperature. And 94 if Taq polymerase can denature, if the Taq polymerase can act at 90 degrees Celsius, which is the denaturation temperature, then 94 degrees Celsius is also permissible. So this option is also not valid. Third option, the PCR reaction will fail as Taq DNA polymerase are not effective enough. This is also not true because Taq polymerase is meant to operate in this temperature and it will work. So let's come to the last option. The PCR reaction would fail as DDNTPs won't allow to incorporate other nucleotides and the extension would stop. So what is DDNTPs? DDNTPs are dideoxynucleotide triphosphates. And in DDNTPs, what happens is, I don't have a figure here, maybe I can draw. Yeah. So if this is your ribose sugar, if this is your sugar in ribose, you will have uh, in ribose you will have two OHs, whereas in deoxyribose one of those O's will not be there. You will only have H's, and in dideoxy ni- nucleotide triphosphates, both the O's will not be there. There will be only be H. So what happens is the preceding or succeeding nucleotide will not be able to bind to any of the sites. So when we discuss that a DNA sequence goes from 5 prime to 3 prime or from 3 prime to 5 prime, this is what we mean by binding. So if this oxygen molecule is not there, then it can't bind to this nucleotide. So instead of DD and TPs, here they are given that they are using a DD and TPs mix. So instead of a DD and TP, what we use typically in a PCR is a just a D and TPs, just a deoxynucleotide triphosphate. So instead of a DNTP, if you use a DDNTPs, the extension won't happen. So the PC extension would stop basically. So the correct answer for this question will be option D. That is the PCR reaction would fail as DDNTPs won't allow to incorporate other nucleotides and the extension would stop. I hope this explanation and the answer is clear enough. I'll wait for another like 5-10 minutes so that I can absorb the answer that was told. And in 2-3 minutes, I can go to the next question. Okay then. Even if you guys don't have any doubts right now, I can come back to this question later and we can solve this question. So I'll move on to the next question. Uh, so second question is this: We are we all are well acquainted with ISRO's successful Chandrayaan 3 mission. On a separate project, ISRO's lead lander sent some samples from Moon. Hypothetically, the samples were sent to a lab at IIT Bombay for understanding the genome constituents of the organisms on the Moon. Dr. Sushila led the project and extracted the DNA sequences from the samples. After sequencing, Dr. Sushila found 4 different types of DNA sequences. On further analyzing, she found the following composition of DNA from 4 types of DNA sequences, supposedly from 4 different organisms, as follows. So we have 4 samples like which might be DNA sequences from 4 different organisms. So what they have mentioned here is how many in each organism they are mentioning how many in the DNA sequences of each organism they are mentioning how many bases of each um, uh, base is there. That is how many bases of A, G, T, and C are there. Adenine, guanine, thymine, and cytosine. How many bases are there? So the question is which of the organisms DNA composition matches with the double-stranded DNA composition of the organisms of Earth? So you just have to choose which organism has the uh, composition of, like as a DNA which matches with the double-stranded DNA composition. So there is a while this concept you would have come across in the first or second lecture of your week two's lectures, and it's an important rule in the formation of DNA formation, and it's a mathematical. It's it is a ratios. It can be called as a ratio or a mathematical formula. So you approach this question if you know that concept or that mathematical formula, you can approach this question easily. So you just need to know what formula is this. I'll give you some time to solve this. So basically, you have a couple of numbers. You just have to work around and see which ones match and which ones don't. So based on that, you have to say which organism will be matching Earth's DNA composition. Any attempts at this answer? Anyone from the five people who are here? Okay, then I'll go on to explain this. So basically, there is this concept in DNA, in the constituent of DNA called Chargaff rule. So there was this person called Chargaff. He invented this rule, and he basically said that the ratio of adenine to thymine bases will be equal to the ratio of the guanine and cytosine bases. So basically, once again, I'll open my pen. So basically, your A by T will be equal to your G by C. So this is the basis of the Chargaff rule. So among this four organisms, you just have to plug in the values of A, G, T, and C, and then which you just have to see which one matches this formula. Which one of these organisms matches this formula. Whichever ones follow this rule, it's going to be your uh, it's going to be your Earth organism. So, which one of these guys do you think that it will follow Sarkar rule? So, if you go by one, A is in organism A, we have 560 by 280, which is 2, but 280 by 560 is 1 by 2. So, it is not correct. 560 in organism B, 560 by 560 is 1. 650 by 650 is 1. In organism C, so 560 by 140 is 4. 560 by 140 is 1, once again 4. And in organism D, 140 by 560 is 1 by 4, and 140 by 560 is 1 by 4. So among all this, B, C and D matches the A by T is equal to G by C. But one more important thing that you need to consider in Chargaff rule is the number of A should be equal to number of T, and number of G needs to be equal to number of C. Yeah, Shambhavi said organism B. So there are two components. A by T is equal to G by C, and A is equal to T and G is equal to C. That also needs to be counted. So in organism C and D, you can see that A and T is not equal, and even in organism D, A and T is not equal. So the right option for this question will be organism B, where we have 560 of A, 650 of G, 560 of A and T, and 650 of G and C. So is everyone else able to follow with me and get this answer as well? Okay then. So uh, I'll quickly move on to the next question as well. So third question is, an operon is a cluster of coordinatedly regulated genes. It includes structural genes, that is enzymes, regulatory genes, activators or repressors, and regulatory sites such as promoters and operators. Consider the following situation of operon. So in this particular operon, this is going to be the situation. Regulatory element is present, which results in operon being on, and mutation of the regulatory gene is to lose function, which results in the operon being off. So the question is, which of the following given statements is correct regarding this type of regulation? So from these two situations, what can you conclude about this operon? So regulatory element is there, which results in operon being on, and regulatory element, something happens to the regulatory element, so it becomes something happens to it, so the operon is off. So it's kind of a positive regulation. Is anyone able to follow with me? So they are just asking which of the following statements will be true. So the first option is genes in this operon are expressed unless they are switched off by a repressor protein. Inactivation of the repressor shall lead to the turning on of the operon. Absence of positive regulatory protein shall lead to turn off of the operon. And inactivation of the repressor shall lead to the turning off of the operon. So which one of will be correct? So I already told you that this is a positive regulation. This is a case of positive regulation. So in positive regulation, if the regulator is there, then the operon will be on. If the regulator is not there, the operon will be off. So it's a pretty straightforward question right now. So the answer will be C, where the absence of the positive regulatory protein shall lead to the turn off of the operon. So mutation of the regulatory gene, just like in indirect sense, it just means that the regulatory protein is not there. So it will lead to the operon being off. The options B and D are exact opposite of the whatever it's intended here, and this is also pretty opposite to the not following the lines of what these two statements mean. So the correct answer will be option C. The absence of positive regulatory protein shall lead to turn off of the operon. So I'll move on to the next question. The next question I think is a bit uh, problematic one where you need to sit and solve the problems. So there is a plasmid map. Consider the given following plasmid map carefully. Rohilla performed a digestion experiment of the plasmid and got the following band pattern after agarose gel electrophoresis. Lane A has one band of 29 kilobase and lane B has three bands of 6, 11 and 12 kilobases. What does the lane A and lane B correspond to? So this is what the question. So they are given this plasmid map. So we all saw in the classes how do we design this plasmid map, and we also saw how to interpret the agarose gel map from the plasmid map. So we just need to figure out what can be like what does lane A and lane B correspond to in this context, which they have a couple of restriction sites like EcoR1, BAM H1, EcoR1 is 2 1 and BAM H1. So among this restriction, and they also have the kilobases between each restriction site. So we just need to find out which of the restrictions sites get cleaved in lane A and lane B. I'll give you around like 3:45 to solve this. We have a lot of time till 5. So I'll give you a lot of time to solve this. At least till 3:40 you can take your time to solve this. It's a pretty simple question compared to the next question. So if you get this question, then uh, you can easily like you can try to solve the next question as well. Okay, then we'll try to solve this question now. So, yeah, so this is a pretty straightforward question. So, the total kilobases that are there in this plasmid are 29 kilobases, and the lane A already says in the question that uh, lane A has one band of 29 kilobases. So, this just means one thing. So, this just means that the plasmid is being cut at only one site. So only one restriction site is being used in this place, and only one site is being cut. So automatically your so the simple way to go around this problem is automatically you can realize that lane A will not be used by more than one restriction site. But here the options say that lane A says double digestion, lane A says double digestion, lane A says double digestion, which will not be possible. So automatically you will rule out options 1, 3 and 4. So the correct option will still be option once again. So the correct option will still be option B only, which is digestion by Stu1, and lane B is double digestion by Stu1 followed by eco R1. But still, let's see what happens when you digest it and in lane B. So in lane B, you have STU1, you cut it here, and then you double digest in with eco R1. So here and here. So you get one fragment which is of 11 kilobases, one base here which is of 6 kilobases, and one stretch here which is of 12 kilobases. 9 plus 3 which is 12 kilobases. So that is what they have mentioned here as well. Three bands of 6, 11 and 12 kb. So this also follows with your answer. So this is the simple way to go through this option. You just go over the options one by one, and then you see which one kind of which one you can rule out easily and which one you can take as your correct answer. So if there are no doubts with this, I will go on to the next question. So consider the following agarose electrophoresis carefully. So they are given this huge electrophoresis where they have different where they have a standard of different molecular weights, and they have different cases. So they have some plasmid, and they have we don't know about the number of restriction sites, and we don't know what kind of restriction enzymes can act on the plasmid as well. So they have HIM3, they have different five cases: HIM3, ECO R1 and HIM3, BAM H1, ECO R1, and BAM H1 and HIM3. So they each enzyme when acted upon on its own or with combination gives you a different gel pattern. So the question is, Preeti wants to interpret the plasmid map from the provided across gel electrophoresis. Help her by identifying the plasmid vector map. So they have also provided four options. You have to choose any one option from these four options. How will you go ahead? So I'll again give you time, maybe till 3:50. Like you guys can take your time, go over the gel as well as the plasmid map, and you can see which one will be this thing. So one takeaway message from the last question that you can see is, if there is one band, if like the number of bands under each case almost always signifies that those are the number of restriction sites being used. So if hint 3 is being under when hint 3 alone is being used, hint 3 enzyme is being acted on, it gives only one band of 34 kilobases. So this means that there is only one hint 3 site almost always. Yeah. So take that ration, think in that way, and see how you can form a meaningful plasmid map. Which one of those will be correct? does anyone have any hints to how you got the answer? Like, so one more thing, one thing that I told earlier is that your uh, number of, if there is only one restriction enzyme, and if it gives only number of bands that it gives, almost always says the number of restriction sites the particular enzyme has. So you can use the options from here and also see which one of them will match which case. Uh, okay, you're saying C, but can you explain how it is the case? Like, okay then, like if I can go into explaining the answer as well. So we'll start with hint 3 here. So hint 3 has only one band of 34 kilobase. So whichever one has only, so there will be only one restriction site for hint 3. And here we can see that um, this one has one, this one has one, this one has one, and this one has one. So all the four options are kind of possible to be the final plasmid. But we'll come to the case of BAM H1. BAM H1 has two bands. One gives a 31 kilobase and one has one's molecular weight is 3 kilobases. So we know that since it has two bands, we know that BAM H1 has two sides of two restriction sites. So which this option D, if you see, there is only one BAM H site. So option D is automatically ruled out. The other three options are valid: BAM H1, BAM, two BAM H1s, two BAM H1s, and two BAM H1s. But let's look at each case. Let's look at Shambhavi's case first, where she said C. So two BAM H1s are there. If if BAM H1 cleaves here and here, then let's see what will be the molecular weights of each of the strands. So it will be 8 plus 8 plus 4, 20, and 3 plus 5 plus 6, which is 14. So 20 and 14. But what we are getting here is 31 and 3, which is not the case. So this can't be the correct answer. So C can't be the correct answer. If we come to the option of B, let's just say BAM H1 and BAM H1 are here. So even in this case, BAM H1, if the BAM H1 cuts here as well as here, we get 3 plus 5, 8 kilobases, and 8 plus 8 plus 4 plus 6, that is around 26 kilobases. But again, we are getting 31 and 3. So that can't also be the correct answer. But check in this case, on option A, if BAM H1 cuts here and here, we have 3 kilobases here, and we have 8 plus 8 plus 4 plus 6 plus 5, which is 31 kilobases, and that's what we are getting here. So even though they give all the other options as well, we need only Heng 3 and BAM H1 to solve this uh, plasmid problem. So the final answer will be option A, which is this plasmid. Yeah. If you try to uh, put all the other cases as well, eco R1 and Hin3, or eco R1 or BAM H1 and Hin3, this will also follow the same pattern. So we can just see eco R1, for example. Eco R1 says that it has 24, 6 and 4 kilobases as the fragments. So if eco R1 is cleaved, we can see what happens. Eco R1 sites are here, here and here. So here, here and here. So we'll get one fragment of 4 kilobases, one fragment of 6 kilobases, and one fragment of 5 plus 3 plus 8 plus 8, which is 24. So that is what we are getting here as well. So is everyone able to get how we are solving this problem? Okay then. So I'll move on to the next problem. So this is a very straightforward problem. I also we discussed this in the uh, recap session. So Ramya wishes to set up a cloning experiment in lab. She is following the steps provided below. What enzyme she need to use for performing step 3? So step 3 we'll just see what happens. So step 3 what happens is basically they're taking gene of interest and they're trying to form the recombinant plasmid. So they're taking the plasmid out and then they're cutting it so the plasmid has sticky ends. So something happens where the plasmid and sticky ends incorporate this gene of interest to form the recombinant plasmid. So what enzyme is the question is what enzyme is being used? And this I already explained in the recap that uh, we join the sticky ends together by an enzyme called DNA ligase. So that's the correct answer. So moving on to the next question. So Rohit successfully transformed the recombinant plasmid into competent cells. Which of the following steps would ensure that the transformation was successful? So question the once again this is a very straightforward question. So someone successfully transformed the recombinant plasmid into competent cells. So they have incorporated the plasmid is inside the cells. So what step would have ensured that the plasmid enters the cell? So there are four options here: cold shock, acid treatment, heat shock, and alkali treatment. So which one of and they have also said like the reasons for each of the thing. Cold shock is to freeze the reactions inside the cell so that the transformation does not happen without hindrance. Acid treatment they use to lyse the cell's protective layers. Heat shock to loosen the protective layers and let foreign objects go inside the cell. And alkali treatment to digest the cell's protective layers and let the internal components come out. And this is also a straightforward thing. So basically you don't want to break the membrane, so your acid treatment uh, won't work, because if the membrane is lysed then the bacteria will not be functioning. Alkali treatment also will, if you digest the protective layers then it won't be functional. Cold shock, if the cold shock can freeze the reactions inside the cell but the protective layers are still intact, so the final option will be heat shock. So only if we give heat shock and loosen the protective layers, our plasmid of interest can go inside the cell, the recombinant plasmid. So if there are no questions, I'll move on to the next problem. So Raman went to Kashmir on a vacation. He isolated different unique eukaryotes from the snowflakes of Gulmarg. He intended to understand the genome composition of the bacteria. For this, he performed polymerase chain reaction to amplify the DNA sequence. After n number of cycles, he found some number of copies of DNA, 10,48,576 copies of DNA. What is the n number of cycles for which he set up the reaction? So they have uh, it's a it's also a straightforward mathematical problem. So you know how PCR works, and like you just have to see how much this copies after how many cycles you will get this number of copies of the cell. So I'll give you like maybe 2 3 minutes to solve the answer, and then I'll go on to explain the answer. You can also use the diagram here, exponential amplification diagram here, to see how many copies is being generated in each cycle. So if you kind of can get any formula for this one, yeah, how many copies can be generated after each cycle, and if you plug the number of cycles, this and that the options are given here. So eventually, if you plug in, you will get this answer. That's the way to go around to finding the solution for this problem. So it's okay. I'll go ahead. So answer lies in this diagram basically. So that's why I keep this diagram from I got from internet. So basically, from one cycle you get uh, 2 power 2 copies. Like uh, after one cycle you get 2 power 1. That is 2 copies, and after 2 cycles you get 2 power 2 copies, and so on and so forth. So the formula for getting the design number of copies is 2 power n. So after after 2 power, so after n number of cycles, you will get 2 power n copies of the desired DNA. That is the general concept. So if you plug 2 power 10 or 2 power 20 or 2 power 30 or 2 power 25, you will get this 108 1048576. So the correct answer is, so it's a, once you know the formula, is a straightforward answer. So after 20 cycles, so if you plug 20 into n, 2 power n 20 will be your 1048576 copies. So that will be the correct answer. So if there are no doubts in this question, I'll go to the next thing. So in advanced problems, you will also have you will also take primary efficiency into account, where it will not be two exactly, but it will be 1 plus epsilon, where epsilon is your primary efficiency. So you will use it, but ideally it will be two, the base. Or if you have a, in exam, if you have a calculator, you can have anti-log base 2, and then you can find the answer. So ninth question, what are the steps in correct order that you should follow while preparing the competent cells for cloning? So this is also a pretty straightforward question. So we just saw about cloning, and we just need to um, accurately predict what will be the correct steps for cloning. So the correct option is, uh, you first incubate it with ice, incubate your competent cells with ice, and then you give calcium chloride salt treatment, and then you again incubate it with recombinant DNA, and then you give a heat shock so that you disrupt the cell membrane, and then you incubate it at finally you incubate it at ice. So this will be the correct way of steps to proceed with cloning. So if there are no doubts, I'll go on to the last question. So Darshan wants to set up a PCR reaction for his gene of interest. The sequence of the DNA strand is as follows. So the template strand, they have given a sequence of the template strand from 5 prime to 3 prime. What should be the annealing temperature? Should be set in the thermal cycle for successful conduct of the experiments? Hint: the forward primer consists of 14 bases. Does anyone have any this thing to the? I mean, normally you have you have specific mathematical formulas as well. So there is like a very proper way of going answering this question, and there is also a very crude way of solving this question as well on an exam basis. So the proper way is that there are formulas for calculating annealing temperature. So once you have a proper annealing temperature calculator, so you annealing, you know that it is based on annealing extension. It basically depends on your melting curve and melting temperature of your primer. So once you know your primer sequence and once you know your template strand sequence, you will get to know what will be the annealing temperature. And annealing temperature also is dependent on the GC content. There is a guanine and cytosine content present in your primer as well as the template strand. Here, the residue number of uh, nucleotides given here are almost like 28 or 29, and your forward primer, they are saying that it consists of 14 bases. So that is almost like only half of the this thing. So your primer is going to bind to the sequence. So this is the template. So your primer is going to bind to the strand only when it is highly specific, because there are 14 bases as well. So once you are the crude way of solving this is, once your bases number of bases is like almost comparable to the template, then your annealing temperature is already going to reduce. So it will be probably the most lowest annealing temperature ever. So the correct answer is 44 degrees Celsius. So almost around 45 degrees Celsius, your annealing is going to happen. So that's all the problems for from the previous week for this question. So I'll quickly go over the answers once again from the first question. So the first question is about PCR. So they gave the reagents for the PCR, and they have given the cycling parameters as well. So they have asked us to choose the correct answer for the among the following options. So here, if we see, the problem here lies with the usage of the dideoxy dideoxy NTPs, dideoxynucleotide triphosphates, which is not the case in PCR. You use a DNTPs. So if you add a DNTPs, your extension won't happen and the extension would eventually stop. So the correct answer for this question is the PCR reaction would fail, as DDNTPs won't allow to incorporate other nucleotides and the extension would stop. So the option is correct, option D. Second question is, uh, they have so in second question we are shown four organisms, and the four organisms are different compositions of adenine, guanine, thymine, and cytosine. So you must be using Chargaff's rules here to find out which one of the organisms which will match the double-stranded DNA composition of the Earth's organism. So Chargaff rule states that the adenine number of adenine residues will be equal to the number of thymine, and guanine will be equal to cytosine because of complementary base pairing, and the ratio of A by T will be equal to the ratio of G by C. So According to all those rules, your organism B will match the double stranded DNA composition of the organisms on Earth. So question number 3 is, an operon is a cluster of coordinatedly regulated genes. It includes structural genes, regulatory genes, and regulatory sites. So they are given a case where regulatory element is present, operon becomes off, and mutation of regulatory gene to lose function, uh, operon becomes off. So basically, it's a form of a positive regulation, and they are asking which one of the following statements is correct regarding this type of positive regulation. So in a positive relation, regulatory element is present, the operon will be off. So then that implies that if the regulatory element is not there, then the operon will be off. So the correct option is option C. The absence of positive regulatory protein shall lead to turn off of the operon. Option D. So they are given a plasmid map and a gel electrophoresis diagram. So based on the plasmid map and the restriction sites that the plasmid map has, we have to correlate the gel electrophoresis diagram and the plasmid map and see which one will be the correct option. So we discussed this, and the correct option is option B, where lane A is digestion base 2,1 and lane B is double digestion base 2,1 followed by eco R1. Question 5. So again, so they this time they are given just a gel electrophoresis diagram, and under they are given it under different conditions, under different combinations of restriction enzymes. So using that data, we have to find out which one of the plasmid maps will hold true for this case. So they are given four plasmid maps, and we also saw that we also solved this using a hint that the number of bands generated under a particular restriction enzyme will almost always be equal to the number of restriction sites for that particular uh, enzyme. So based on that and based on the band size that each enzyme gives, especially in the case of hint 3 and BAM H1 and eco R1 as well, we came to know that option 1 is the correct answer. So option question 6. So in this, so someone sets up a cloning experiment and they are saying that some enzyme needs to be used in a step where the plasmid is with sticky ends forms a recombinant plasmid. And we know from this, from the recap as well as from the classes that a plasmid will be conjugated to a plasmid will be conjugated to your gene of interest through an enzyme called ligase, which helps to bring together them. So the correct option will be option D, DNA ligases. Question number 7 is, uh, Rohit successfully transformed the recombinant plasmid into competent cells, and which step will ensure the transformation was successful? So they are given four options cold where the options include cold shock, acid treatment, heat shock, and alkali treatment. So acid and alkali treatment we can't use because we can't disrupt the cell's protective layer. We can't like destroy the cell's protective layer. Cold shock we can't give because the cell's protective layer needs to be disrupted a bit so that the plasmid can enter. So that the final correct option will be heat shock to loosen the protective layers and let foreign objects go inside the cell. Question number 8 was also pretty straightforward. So they are given the number of copies of the DNA. Using that, we just need to find the number of cycles of PCR. So using the formula 2 power n, we can find out that the correct answer is 20, option B. Question number 9. So we just have to figure out the correct flow while preparing the component cells for cloning. So the correct option will be option C, option A, ice incubation, calcium chloride treatment, ice incubation with recombinant DNA, heat shock at 42 degrees Celsius, and final incubation at ice. And option 10, question number 10 will be someone wants to set up a PCR reaction and see they are given a template strand sequence and they are told that what they are asking what is the annealing temperature we should set, and they also hinted that the forward primer consists of 14 bases, which is like half the number of bases that the template has. So in those cases the annealing will happen pretty easily. So the yeah, annealing will happen pretty easily, so we can set the lowest possible annealing temperature at around 44 degrees Celsius. So these are the questions for week two for the previous year. So till so that's all from my side for this TA session. So after this, from time is 4:10. So till 5 o'clock, which is the typical end of the session, we can have a doubt session where you can ask whatever doubts you have. So if there are no doubts, we can see you next week. Uh, my TA sessions will be on the same every Saturday from 3 p.m. to 5 p.m. So um, till the end of 5, I open this room to doubts. So if there are no doubts, further doubts, no one's in the call. So thank you everyone, and I'll see you next week, the same Saturday, next Saturday, that is 8th February from 3 p.m. to 5 p.m.